Thanks, and welcome everyone to another chapter of today's and tomorrow's exciting Planet Fest 2021. My name is Britney Schmidt. I'm a professor at Georgia Tech, and word on the street is soon to be Cornell, which is kind of exciting. I am a person obsessed with life on this planet and other places, and I'm really excited to be joined by others who have similar interests. So we're going to kind of go around and I'll introduce our panelists, and they can tell you a little bit about the techniques and the technologies that they use that are related to the search for life. And we've got members who are thinking about it on the Earth and on the Earth's history. We've got people who are stretching that to Mars and to our solar system, and then others who are looking much, much farther beyond into other solar systems. So we're going to kind of run the gamut here of people obsessed with finding life on other places. So let's start as close to the Earth as we can. With Dr. Betül Kacar, good friend of mine, professor at the University of Arizona. Betül, would you like to tell us what you're into, what you're working on, and what you're excited about in life detection?
Well, thanks for having me. You said it. I am obsessed with at least figuring out the possibility of finding signs of life elsewhere in the universe. And I start from what we know, that's life that we have on Earth. Earth is a dynamic planet. That is great. That means that there's a lot of chemical potential here, but it also makes it difficult to trace the first chemical steps of life. And my group is trying to understand that chemical potential that once harbored on this planet, and how biology, how life itself integrated that into its systems. So we try to use today's biology and reverse it to understand what happened billions and billions of years ago. If it sounds like a molecular time machine approach, because it is, in some ways we try to travel back in time and resurrect ancient DNA molecules to understand how the first steps of life were impacted by life and the environment, and how the environment in return impacted life. And then once we resurrect these ancient signatures of life, we then try to connect them to the data that we receive from out there. So it's a really scale problem. How do we connect a molecular scale phenomena to the largest scale possible, at the stellar scale? And that's in a nutshell what we do.
So literally just studying everything. So let's take it from the Earth record to Mars and to other planets, and even to the idea of how to rethink what a biomarker or evidence of life might actually be on another planet. So Dr. Heather Graham is at NASA's Goddard Space Flight Center, and is an expert there in laboratory and field methods and the paleo history of our own planet, as well as trying to detect life in all of its forms and other planets. So Heather.
Hi, yeah, thanks so much for inviting me, Brittany. So I'm an organic geochemist, which means I'm really concerned with organic chemistry as it's preserved in the rock record. So that means that when I think about life elsewhere, I'm really using a lot of the techniques that we've developed and perfected for looking at life in distant time here on Earth. Astrobiology really has a strong footing in paleobiology, because for ages, paleontologists have been looking back in time here and trying to figure out how life worked back then. And we can use a lot of those same conceptual models and even tools. A little bit more than just looking for particular kinds of life, though, I'm really interested in the possibility space for life. What are the most basic needs that need to be met, and how can we extend our ideas about life as we see it here to other models of living that might be possible with other biochemistries? That's broadly what we call agnostic biosignatures, where we're being agnostic to the biological system. We're not necessarily assuming that there's heritage to life here on Earth. And in the group that I'm in, we use a number of techniques, mostly chemical, which is good for me since I'm a chemist. We think about molecular complexity, surface complexity, patterns in chemistry, and isotope fractionation to tell us something about whether or not a signal we see is life. And I often like to tell people that we're doing less biology detection and more physics detection. We're trying to find evidence that some signal, some observation that's usually chemical, wouldn't just happen kinetically, that it needed some little bit of extra energy. And that is probably the most basic way you could describe life, is that little bit of extra energy that creates new conditions that wouldn't be expected with just physics.
Which is kind of amazing, that we can think about the molecular tools that Betül's actually engineering in the lab, think about how different chemical settings would allow those to reorganize into different things. Like might be DNA-like, but would it be called DNA? Would it use the same backbone? Would it do the same things? We'd still want to know it was there, which is kind of amazing. Other signals, I liked that Heather used the word signal, because we all talk about signals in our particular research fields, and we mean slightly different things. But one of the really exciting signals that we're really searching for in using modern telescopes is the signal of atmospheric gases or atmospheric indications of life on another planet, or even just at this point in time of a planet that might be kind of like our own. So let's go over to Dr. Tiffany Kataria, who will tell us a little bit more about the search for habitable exoplanets and eventually life on them.
Yeah, thanks, Brittany, and thanks for the invitation. This is a really great group of women to chat biosignatures with. So my research specializes in actually primarily understanding weather on exoplanets in general. And when I say exoplanets, I mean planets orbiting other stars, like the planets in our solar system orbit the sun. There are over 4,000 exoplanets that have been discovered so far via various techniques, which I can talk about if you have questions. But spanning the gamut from very small planets, even smaller than Mercury, to planets that are larger than Jupiter. And the challenge is we really don't, whereas in the solar system, you have the benefit of sending a spacecraft there, or getting in situ or on-site measurements so that you can really apply what we would call a ground truth, or a really basic understanding of what these things are made of, and corroborate that with models and with laboratory measurements like Betül was describing. In exoplanets, we really don't have that luxury. We really have to rely on these telescopes to get spectroscopic measurements, so measurements with wavelength or over a prism of light, and try and disentangle those signatures, getting a single spectrum and understanding, is that from the atmosphere? Is that from a planetary surface? Disentangling all of these effects into what essentially is a global measurement. And so this is where theoretical models, such as the ones I use, come in very critically. So primarily I use three-dimensional models to understand how the weather on a planet might circulate, how the winds might blow, what the temperatures might look like, and what that might dictate for conditions on a Jupiter-like planet or an Earth-like planet. So what are the best conditions for that might be conducive to life? You might have heard about the Goldilocks zone, or the habitable zone, is this construct of understanding the distance of a planet from its star and what that might mean for the temperatures that might be experienced by the planet. It's being hit by incident radiation from its star. But what does that mean if the planet is not actually made of nitrogen like Earth, but maybe made of carbon dioxide or ozone, or something completely exotic? What are the context of, what is our understanding of life in that context? How might life arise in that sort of context and persist, such that telescopes, which are searching for light, from many light years away, from thousands of years away, consistent or persistent enough so that we might be able to see it. So that's really the challenge with exoplanets. And my background's actually in planetary science. A lot of folks who work in exoplanets come from an astronomy background, but I really think, astrobiology is a good case of this as well, but exoplanet science in general is really it's important to have some interdisciplinary collaboration and communication between not just astronomers who look for these objects in the sky, but planetary scientists like the ones I'm looking at here, to really understand what are the physical processes that would bring, whatever's happening on the surface of the habitable planet to be detectable by an atmosphere or by a telescope.
It's kind of an amazing question, is how fundamentally does a planet work, right? It's actually a question that we have about biology on this planet is, was it a part of life? Or part of the planet? Did it arise from the planet? Was it there because of the planet? What are those interactions? It's kind of a really interesting kind of challenge. And talking about signals, of course, I've got my background RIP, our favorite space telescope, or my favorite telescope. So this is the Arecibo Observatory, and you can see the ears of my cat here. I have a kitten from Arecibo. But the signals I wanted to share with you guys that I actually look for are a little bit different. So I'm kind of an ocean world person, and I'm interested in thinking about the types of planets that we may not actually be able to really effectively detect, or maybe we'll detect them, but the signature of life is a little harder to get at. And so the ocean worlds of the outer solar system, and probably moons around planets that Tiffany is going to detect one day, are these ice-covered ocean planets, and they have another story to tell. And so one of the things that we're doing, so we've seen a couple examples. So we're using telescopes here on Earth. We're using Earth atmospheric models to be able to understand other planets, using the geologic record and the biologic record of the Earth. And we're trying to use actually physical environments on the Earth to try to understand these places. So in the back here of this is a place I spend a lot of my time, which is a big yellow tent in Antarctica. And the robot sitting right there is Icefin, which is a robot that my research group and I put together that is a kind of a prototype for the kinds of things that we'd like to one day do through the ice shells and in the oceans of ice-covered planets. And so we're actually trying to figure out what parts of the ice can actually be a signal of life down below, and of what's happened in the history of an icy planet that actually makes it habitable. So Icefin's actually about not quite 2 of me tall. I got a little bit more than 2 of me tall, about that. And so it's kind of a prototype of how we explore. So we're all using these different tools of exploration that we can access here on Earth, and then thinking about what the next steps are. But that kind of, I liked what you ended with, Tiffany. I was going to go around and just ask the panel. It's a question that we always get asked, but it's really kind of a personal thing, is what do you guys think about the word astrobiology? What does it mean to you to be an astrobiologist? So let's go to Betül first, perhaps.
Well, it's a pretty, well, where do I start? So I love being an astrobiologist. I think it's really changed the way I think about life. I never thought that I've been actually in this field for about a little over 10 years now. And I had a background in chemistry, and then I did my PhD in medical school, studying biophysics and biomolecules. And from there, I transitioned into studying life as a planetary phenomenon. So who would have thought, right? From studying human systems and human bodies and how our body responds to different pathogens, transitioning into studying life as a planetary scale phenomenon. And this is really amazing because, as I like the way NASA puts it, that we are not making a fruit salad here, but it's a fruit smoothie, that you know, we're combining everything, and it's a good thing. So the fact that we are all you know, so connected, and we see life as one phenomenon that is you know, studied using different tools, different you know, minds, and really diversity. There's, I think, a really heavy diversity in this field in many ways, and I think it shows in the way that we think. Especially considering that the field itself is not too old, but the fact that we are asking one of the oldest questions that were asked by humanity using these amazing tools gathered by different disciplines is very inspirational to me, that none of us know our places, right? It's a good thing. We don't know our place, and that's a great thing. And I think that, you know, asking these big questions and chasing them gives you that sort of boldness that you dare, and you know, why not? There's one life, why not we ask the biggest question possible? How did life begin? How did it start on our planet? How did it flourish here? And can we find its signs elsewhere in the universe? So I think it's a pretty inspirational thing, and I feel very fortunate to be in this field.
It's awesome. Tiffany, what does being an astrobiologist mean to you? What does the term mean to you? How do you use it in your daily life?
Sure. Well, thank you first for calling me an astrobiologist, because even traditionally I don't consider myself one, because my PhD work was primarily, as I said, in using these 3D weather models to understand climate on planets. But primarily they were for hot Jupiters, these Jupiter-sized planets that orbit very close to their stars. I'll give you the takeaway. The background behind me is actually an image, an artist's rendition of a hot Jupiter. So that's what Heather asked me initially, thought it was Earth. But this is really the challenge here, right? This is based off of the fact that this planet had a reflected, was measured in reflected light, and it reflected essentially blue light. And so they inferred that this planet is henceforth blue. So that was primarily, you know, what I continue most of my research in. But lately, in thinking about what the next generation of telescopes or space-based telescopes are going to look like, what comes after the James Webb Space Telescope, which will hopefully launch this fall, what comes after the Roman Space Telescope, which launches in roughly 2025. What are the next generation of telescopes, and how might we be able to characterize life on an exoplanet, and not just find it, but really understand it? And that really takes a lot of you know, the observations themselves are only one piece. The ability to understand the observations you have are only as good as the models or the theorists that you're working with. And so to me, that's astrobiology. Some of my more recent research work actually goes into understanding how microbes on an Earth-like planet might exude gases that you might then be able to detect in an exoplanet. So I'm working with a team of astrobiologists, or more card-carrying astrobiologists. You know, they do a lot more laboratory work in growing actual microbes in the lab, and then understanding, using the theoretical models that I'm more familiar with, how those might couple together, because really it's understanding the system as a whole. So I think, to me, that's maybe a key point of astrobiology, is understanding the system as a whole.
I can really relate to what you said about, oh, thanks for calling me that, because I don't actually know sometimes what term to use with people, because yeah, it is really, astrobiology is this field of so many different types of people and so many different backgrounds. So I tend to use a different term depending on where I'm at. So I'm a geologist, a geophysicist, an ice person, an astrobiologist, a space fan, whatever it is. So Heather, how about for you? What does astrobiology mean to you? How do you use that term?
That's, you know, it's really interesting. I'm kind of like you, Tiffany. Like, I didn't know if am I one? Do I get to be one? Because I think so much of my background came from looking at the past on Earth, and not even like super far back, like the Cretaceous. Like, we would recognize most of those things. And so that's a lot of my training. But I think what I realized as part of that, and what makes astrobiology unique and why I gravitate to this field, is it really opens up the search pattern you're allowed to use. When we think about looking at the fossil record of life on Earth, we're assuming a lot of things, you know, mostly about physical form and metabolisms and things like that, about what you can expect, just because we know Earth's life has a deep history that is interconnected and inherited by all of us, all these critters. But when you go into astrobiology, you get to open that up, and you can do astrobiology here as well as other planets. We can ask questions about really strange places that don't seem to harbor life and say, oh gosh, could there be something slightly different that doesn't match with our search criteria, and that's why we're not seeing it the same way we do on another planet. And so I think that's what I love about thinking about astrobiology. I think the other answers were super, thinking about systems level knowledge, of course, but just that allowance to let your scientific creativity really flow and think about, like, what are actually the hard limits on any of these processes.
So I'm going to kind of exploit that. We were talking about what would we know based on what do we see, and using this whole bunch of lines of inquiry, system science. We throw that term around. For folks that aren't really familiar with the concept of system science, it really is, if you think about it, if you described it, it's kind of like the cake analogy, right? If you put flour and sugar and eggs and stuff and you just set it in a box, it wouldn't really be a cake. But if you were detecting those ingredients, you could think about it as a signal of a cake. But it's how it's mixed together, in what order it comes together, and in how hot you baked it, that makes it delicious or complete tragic failure, which I know we're all learning, living at home, cooking for ourselves a whole lot more than we used to. But I want to let's go further into that. Why is it so hard to find life, Heather? Just a little question.
Oh, that's all.
Tiny question.
You know, that's interesting, because when I think about finding life, you know, it's not always easy here. especially if you think about the geologic record. And so you do have to use those search patterns a lot, so that when you do dig into some rock and find some, strange molecule, can you compare it to something you may have seen in a living system? Or does it seem to be absolutely impossible for it to have arisen just through physics? And that right there would be kind of the basis of an agnostic biosignature, for example. It's just like, how impossible is this thing? Why? And because I don't see it in other living things. So I think, when we think about other planets, we're dealing with a lot of time that may have happened in between an extant life system, a living one, and when we're observing it now. And so that's something else to contend with, which of course, really takes me back to kind of my roots scientifically.
Betül, when you think about this question about detecting life and why it's so hard, I think there's a lot of humility that we have to approach the search with, right? Is just how much we don't know. And I was wondering what your thoughts were on that. Why is it so hard?
Well, so everything that we know about life comes from one sample that are from our own Earth. And I said, as I said in the beginning, Earth is a living dynamic planet itself, which is great because there's a lot of chemical potential here. But at the same time, the earliest records of life are under continuous risk of being overwritten or destroyed. So going back to say Mars, what makes Mars so appealing is that because it's been in a comparative state of arrested development in a way, right? So there's much greater chance that early planetary systems or even prebiotic chemistry that was harbored on that planet was protected. So the fact that we are not we're just getting started to access this sort of past information that may be recorded on different planetary systems. And we're really at the actually really first steps of, I think, truly searching for life. So I don't think that, I don't think it's going to take us long once we start developing our understanding of the possibilities of what sort of planetary conditions and environmental conditions and chemical conditions are needed for life to start to finally find it. So I don't think we're limited in our technology per se, but our understanding of the possibilities.
Yeah, I think it's really amazing. It's why, the work you're doing to understand the history of life and kind of back engineer it is so important. If folks saw I changed my background, I've got a few fun backgrounds related to life detection. These features behind me are actually modern day versions of what we see as some of the earliest geologic records of life on this planet. They're called stromatolites. These are in Western Australia in an environment called Lake Thetis. And it's kind of a crazy environment where water that used to be part of the ocean got kind of sequestered, and now it's kind of evolved slightly. And so these are communities of organisms that are just living in a kind of a cool way. But if you walked up to it on the shore, you'd say, this is a rock, right? So it is hard because life doesn't do what you expect necessarily or what you really want it to do. Tiffany, obviously you've got probably, your field specifically has one of the hardest questions, right? But why is it so hard for thinking about it for exoplanets or for telescope development?
We need more data. It's my one line. That really is the challenge. Like I said earlier, we're really relying on the capabilities of our current telescopes. It's really the best example of trying to make lemonade out of lemons. Not to say that these telescopes are by any means lemons. It's just that none of these telescopes so far to date have ever been designed to detect signatures from exoplanets, let alone try and find life. And so, it's really about using the facilities that are currently available to us and trying to get the best information that we can. And so that's why I was saying for the next generation of telescopes, we're really thinking really hard about, what are the best wavelengths that we, if we want to detect methane or water or ozone on an exoplanet, what are the best instruments that would be capable of doing those things, and for what types of planets should we be prioritizing our search for life on. So as I said, there are over 4,000 exoplanets that are found, and they're not only different types of planets, but orbiting different types of stars. So from stars that are more like our sun to stars that are maybe half as cool, They may have a lot more flaring or stellar activity, which might be, you know, really kaput for life on a planet. So these M dwarf stars I'm alluding to, they're much smaller than our sun. TRAPPIST-1, for those of you in the know about exoplanets, that is a system of 7 roughly Earth-sized planets orbiting an M dwarf star, which is, hooray, Earth-sized planet. But what does that really mean in terms of actually finding life on that system? It's really about understanding, you know, how might a star that's constantly flaring radiation, are these atmospheres going to survive? Is life going to survive in an environment like that? And so, you know, circling back to what Heather said, it's really about expanding your thinking about pathways with which life could arise. It's not just about you know, Earth is our data point, similar to, you know, everyone else on this call, right? We're only relying, that's our only data point too. But then expanding that to even more extreme conditions than what we see on Earth. I'll just close with an example. I was actually just, you know, the thing about exoplanets is you do a Google every day and something new will come up about something exciting someone's working on. And so I just saw a press release of a group using laboratory measurements, actually looking at iron oxides at higher pressures to understand how that might behave in certain conditions. And so, you know, a lot of terrestrial exoplanets that have been found so far, they might be 5 times more massive than Earth. And so what does the interior structure of a terrestrial exoplanet look in that environment? And so if you increase iron oxide and combine it with other materials you might find in an Earth interior, they may not mix the way you anticipate they would like they would on Earth. They might have a different viscosity, different rock properties. And so, you know, starting from the very, very basic fundamental things, and then how does that scale out to life? It's a really, really, really complicated problem. And it's one thing to understand those processes. And again, another thing to actually, what are signatures that we might be able to find that would indicate those things are taking place? They're very complex cakes.
Absolutely. So that's a great also a starting point for the next question I was going to ask, and we'll start with Tiffany. What is, if you had to pick one mission that you either are excited about that's being proposed right now, or your dream mission that you would get to send, you can answer one or both of those. What mission would that be? And just a little bit about it. And
sure. Yeah. That's if we had all the money in the world, if you know, really at the end of the day, what exoplanet science benefits from are telescopes, light buckets. So the bigger the telescope, the more light you can collect. Ultimately, so most of the planets that have been discovered so far have been using indirect techniques. That is the transit technique, which to use a prop that I have, oh gosh, okay, you can see it here. So imagine this is a sun or a star. You're imagining a planet passing in front of the star, behind the star. That's the transit technique. Radial velocity, you're measuring the gravitational tug between those two things, but you're only inferring that the planet is there based off of that sort of signature. So ideally, you want to take a picture, which is what we call direct imaging. And so that's primarily been done for much larger planets, even larger than Jupiter, around very, very bright stars, much hotter than our sun. And so ideally, we would like the techniques to be able to image Earth-like systems, so a sun-like or a sun-like star and an Earth-like planet. And so that's really a challenge of contrast, because the sun, the star, is going to be, you know, thousands of times brighter than your planet. And so ideally you want to block out the light of the star so that you can see the planet there. And so that requires very fine optics, very large collecting area, so you can get a lot of photons, a lot of light, so that you can really make out the planet that's there. And you want to look at it over the course of an orbit, ideally, so that also requires time. So you're asking this question at a very good time, because in the astronomy, the astrophysics community, we're currently undergoing what's called our decadal survey. And so every decade, the community comes together to decide what are the next priorities for the following decade. And so, in fact, Brittany and I were directly involved with some of the activities related to that, which was very fun. And so with that came a lot of concepts that people brought forward. So what would that next telescope look like? And so actually 3 of the 4 large flagship missions, so the big space telescopes that you might put into the sky, each of those have focus on exoplanets and finding life. So 2 of them, LUVOIR and HABEX, these are 2 direct imaging concepts concepts. I should say concepts. They have not been selected, but their goal is to do that just as I was describing, imaging Earth-like planets around sun-like stars. But again, taking those observations, coupling those with models. So, it'd be great if we had the funding for a telescope like that. I don't know if we do, or maybe we do over many, many years. And it's also a challenge of technology, just getting it to the point where those telescopes are actually capable of doing what we'd like them to do. And yeah, I mean, in an ideal world, that happened tomorrow, but we'll see for the next decade.
Yeah, we'll have to work just a little bit harder on day of action for sure for TPS. No, it was really a fun process to be part of the decadal surveys. Is actually how the scientific community kind of gets its priorities together as a group and then kind of moves forward for the next 10 years. Spacecraft that are launched tomorrow probably were in development for 20 years before they were maybe even selected in some cases. And so we've been working for a long period of time on the concepts that are just being decided between with this decadal survey. Betül, how about you? What mission that's been proposed, or what mission would you propose, or what would you want to do as our next step or as an ideal step in the search for life?
Well, first of all, I want to say that any mission is better than no mission. So I wish that we didn't have to choose from any of these things. We will turn any of these things into lemonade. There are really smart people behind all of these ideas. It takes such a long time, to propose, to come to the level of proposing that mission. There's decades and decades of work, even, years, and a lot of smart people. So I wish that we didn't have to even choose. So if I had to choose, I would say send all of them, okay? But well, realistically speaking, I think I would really want to understand the profound questions that will allow us to understand, again, life and the likeliness of life to exist elsewhere in the universe, especially considering that we're still in the first steps of searching for life in a very systemic way at this point. So the question is then, how can we understand the chemical potential of our solar system? So aside from Mars, I think Europa and Titan are great places because they will allow us to understand, going back to the proto-Earth, so how did the proto-Earth and the chemistry in the proto-Earth may have allowed life to flourish? Now the question is exactly, yeah. And now the question is, how did the chemical conditions of the proto-Earth allow life to transition, right? There's a big how there that we still don't know how that happened. So I think I will go to, do we need to look for a planet that's like ours? Do we need to look for a planetary system that's like ours? The moon and the planet system itself seems to be pretty unique. Do we need to look for a solar system that's like ours? So there are three different questions. It's like a matrushka doll, you open and there's another question and there's another question. It keeps going and going. So that will, I think, all brings us to the proto-Earth chemistry conditions. So I think we need to understand our solar system really well. And of course, exoplanets are great, and again, I would love to see what's out there. But I think that we really need to understand what made life possible in our solar system at first place.
Absolutely. Excited. Thanks for the Europa shout out. Had to put up the next background. Heather, let's go over to you. What are you excited about, either the next mission or an ideal mission for you?
Well, I'm kind of like Betül. I just don't feel like I could choose, right? Like, oh no, don't make me choose. But that's it, I will. And I would definitely, sorry, Tiffany, I'm really focused on our solar system, mostly because I'm a very in situ scientist. I'm a field person, I'm an experimentalist. So I want to be able to go to a place and do an experiment. And for me, the ocean worlds are really exciting. I won't choose Europa and Enceladus, they're both cool. And part of what I really like about being able to go to a planet like that, if we could access the water there, is that it would give you a way to interrogate, to look at both the soluble components of that water and particles in that water, and understand the chemistry of particles there. Is it a cell-like thing? Is it not? As well as any other sort of chemistry you might see there. So just the diversity of different kinds of data you could get in a system like that is really enticing to me. And I say that as a geologist.
I completely agree with you. I'm, of course, biased a little bit. Don't tell Europa, my favorite planet is actually Triton, but I think about Europa, and that's kind of this kind of picture, like what Heather was just talking about, putting robots underneath the ice is the dream goal. So for me, that's kind of my, what I think about. We for the last 2 years, a team of like, 40 or 50 students, and I and a couple of my colleagues at Georgia Tech have been working on a concept study for a mission called Vern, which would be kind of a melt probe profiler with an ocean profiler on the end. So you'd kind of like melt in and explore the ocean and the ice shell, but then also kind of profile the upper ocean. I mean, I think in the long term, I'm like everybody else. I want to be in the submarine underneath the ice and going for it. That's clearly not going to happen in my lifetime, but it's really exciting to think about. But it's also been just so much fun to be a part of exploring all of these planets. And we all have our favorites and our favorite kinds of things, but I wanted to make sure that we just talked about all of the excitement in the solar system and beyond, for different planets and different types of missions. And telescopes are a big part of it. I did my PhD on Hubble, so like the idea of super Hubble is amazing. We're like kind of fighting over, oh, is it going to be LUVOIR, which is 16 meters, or is it going to be HABEX, which is like 4 meters, and Hubble's like 2. I mean, it's just, they're amazing, the difference between those mission scales. It's just incredible. So what a fun time that we live in right now. So I'm going to go just really quickly. We are here at Planet Fest, and Planet Fest is a planetary society tradition. We try to have a big event celebrating any of the landing events, and we're of course celebrating or pre-landing of Perseverance on Mars. So let's just highlight our favorite thing about Perseverance, and then we'll take some questions from the community online. So we'll start again. Heather, if you wouldn't mind, what's your favorite thing you're looking forward to for Perseverance?
Well, the thing that I'm most excited about, of course, is sample return. I want those rocks back here so I can
answer too.
But I do have a second answer, which is actually about Perseverance, and it's super nerdy, and I invite you all to learn more about this. And it does go to a question we had, which is witness tubes and blanks. I love the amount of effort that's gone into making sure that we can track any sort of contamination and understand how the different tools that will be used behave with other physical properties of these things that are sent with it. So I know that's so nerdy. Oh, blanks, that's so exciting.
No, but what is it? Can you give us a little more detail? What's the witness tube?
So they're sending empty tubes, just like the ones that will be filled with sample, along with it, and that will come back, and then we'll understand if there were any processes that affected the integrity or the chemistry of those tubes that might have influenced the samples during the time of collection and return. And then the witness material that they're having that's a drillable blank is something that we know what it is, and then they'll take a tube of it so that we'll understand, if there might have been some sort of chemistry that happened as it was drilling under those Martian conditions, and be able to track all of the changes that might have happened to that well-known material on its trip there and back.
Yeah, that's the kind of detail that you don't hear in like a press release. NASA's super excited to send blank tubes to Mars to see if we don't screw it up. It's a really important part of it, right? There's so much to each one of these fields and to each one of these missions. You have to try to outthink yourself so you don't make a mistake. Now, that's a fantastic example. Betül, how about you? It looked like you were really enthusiastic about that effort as well.
Anything that encapsulates time, I love that, right? Anything that just sort of arrests it, I like that. So I mentioned in the beginning too that we are talking about a planet that's in a sort of arrested development stage. And as someone who's interested in origin of life, of course, I'm interested in learning about the prebiotic chemistry that will come from this mission. So the nature is a better chemist than any of us. Right? Let's we're very smart, but not smarter than nature. So I think it will be very interesting to learn from these preserved conditions and how prebiotic chemistry existed on this vast field. It's the best field you can ask for, I think. So I'm really excited about integrating what we learn from the chemical conditions of Mars in our studies when we try to understand how life originated at first place.
No, I think that's absolutely fantastic. Whatever samples come back, we always talk about the non-detection. As much as it's not going to make as many headlines, is just as important, because it tells us what was wrong and what we thought before, or what we missed, or it tells us something else about the planet that we didn't know to expect.
Yeah, we want to be surprised. I want to think like, oh wow, I never thought about that before. I feel really stupid. I want to feel like that.
Yes. It's a big part of being a scientist, being like, oh, I was wrong, and now I can say why.
The entire time.
Absolutely. So Tiffany, it's got to be exciting because you're sitting at JPL, the center of the action. So what are you looking to for the landing of Perseverance?
Sure. Yeah. I mean, I actually was on lab at JPL for a brief thing the other day and got to see some of the setups that they have, basically for no one. No one's really on lab, such as the times we live in. But yeah, I mean, certainly I would have loved to be on lab for the whole shebang. It's a really fun event. Any of these landings, any of these launches, really, it's a really reaffirms kind of what can be accomplished when a team works together and is successful. So it's really even for people that aren't on the mission, like I'm not involved in Perseverance in any way, but it's very gratifying to be a part of an institution like NASA that can do these dare mighty things, and so obviously, yeah, being at JPL, excited about the mission, but and you sort of stole my answer, Brittany, but I'll just reemphasize, understanding how life might not form in an environment is actually really important. And so, especially for exoplanets, understanding all those, negative pathways, how life could probably not flourish is just as important as figuring out how life might arise. So ruling out those false positives, for an exoplanet, you might detect a gas that you might guess is from life, but maybe there's another pathway that actually can generate the gas that requires no life at all. And so understanding how that might take place on a planet in our solar system is important for understanding exoplanets. I'll also just say I'm excited about the helicopter concept. I'm excited about its ingenuity. I mean, I'm just demonstrating that sort of technology on a planet other than Earth is really exciting. I really hope it works. But that could really open some doors for some future concepts in the solar system.
I think that's it's going to be a pretty spectacular event. No matter what happens, we're going to learn a ton. One of the things we always learn is that we have more questions, right? So a few of the questions that I saw early, actually, Heather, this might be a good question for you, but I wondered about there's kind of this. It's not really a controversy, but this kind of relates to this idea was the Viking experiments, right? Like the labeled release. And so could you kind of explain how scientists in the community think about the labeled release experiment and its results? And if Betül, if you wanted to jump in as well.
Yeah, I think the Viking results are such a great example of good science happening. Primarily because we're all still talking about it. We're all still doing experiments to try and interpret the data, but also we took that data and we didn't just go with one observation. We had a bunch of experiments that we designed to work together as a suite, and the conclusions that were made weren't just based on one thing, but the conclusion that there wasn't life found is because that's what the preponderance of evidence showed. Most of the evidence showed that. It does beg the question, what was different about the labeled release experiment? And for those of you who don't remember, that was the one where they took a sample of the soil or regolith on Mars, and they added a kind of nutrient solution with radioactively labeled glucose, so sugar, as well as some other nutrients. And the idea was if there was any critter in there that was just cold and hungry, that nutrient solution would wake it up, it'd eat, and it would respire a radioactively labeled gas. And that was observed, which is very exciting. Oh my gosh, it's life. Except that when they would re-add more, you didn't see that. And if it was biology and you added more food, you would once again see metabolic activity. So that was another piece of evidence that led us to believe, well, that might have been just a chemical reaction instead. And I think Betül could probably speak a little bit more to the fact that the growth curve kind of thing that you would see with those kind of respired gases didn't look like biology either. It looked like chemistry.
And so there are actually many layers in this concept. The first one I want to touch is actually following up on Tiffany said, that we are looking for life, but convincing ourselves that there's no life on this system or planet is as hard as finding life itself. I think for a really long time, we thought that finding life is, I don't know, running into a tulip in the desert. I live in the desert. I look for tulips. It's hard. And, and then we realized that, especially through these experiments, no, it is not like looking for a really colorful flower in the desert. It is going to be really, really harder than that. It may look like the desert itself, because guess what? There's a lot of biology in the desert, and there's a lot of life under the surface. So, and I think this also goes back to our, there was, I believe, questions about this too, the planetary protection, and how do we make sure that we did not contaminate that planet with what we brought in. And especially these days, it's just a remarkable time to, for biology. We can sequence so many genomes. The metagenome technology is just amazing right now. It's just an incredible time to be a biologist, and it's an incredible time to be an astrobiologist. It's an incredible time to be an astronomer and a planetary scientist. So it's an incredible time to be a scientist, period. So, it's really, I think, great that we need to use these technologies, and we increasingly will, to make sure that we did not, carry anything with us. And I think the Viking experiment was great in the sense that, we made at least me think that, wow, it's a very complicated problem, and we need to think of it in a very, very multidimensional way.
Yeah, I think the concept of experiment design, how do you design an experiment for a place you've never been, is important. And that whole suite of experiments and the detail Heather was just talking about too, about sending a blank and sending that along. And Betül, you mentioned planetary protection, so I'm going to riff on that for a second, just because it's a question that folks have asked. And one of the things that we used to think, at least kind of 20, 30 years ago, so astrobiology is like, depending on how you catch it, is like 50 to 70 years old-ish, right? So it's actually not a very old science. But this idea of planetary protection has two different factors. One is, if we detect life on another planet, and if we use the cake analogy, it'd be like if we saw, eggs and sugar and whatever, okay, that's life. If we detected it on a planet, we'd like to make sure that we didn't actually bring the cake with us. And then the second part of it is our responsibility not to go and mess up future science experiments or populate a planet with microbes that could live there. And there's kind of there's a lot of facets to that. But I think it's just an important difference that Betül just mentioned between the way planetary protection was thought about even 10 years ago, and what we can do now. One of the biggest things that we can do is we can clean the heck out of a spacecraft in a lot of different ways. Actually, using food science technology is probably going to be the frontier in how to do this. We've been doing it one way for like 50 years. We're going to do it new with these upcoming missions. But the other one is just take the ability to look for Earth life with you. Mm-hmm. We know so much more about the genome on this planet that if we find DNA that looks like something we can genetically trace to the history of the Earth, it's probably not something we just found that's new. And so that's a new field. I don't want to spend the whole time thinking about that, but it's really kind of interesting. But this kind of gets back to this other question. So there's a really neat question about how are these discoveries of new planets outside of our solar system affecting how we think about life, but I think also just about our own solar system, right? We've learned a lot. And Tiffany, I'd love for you to speak to this about what is and isn't required in the formation of a planet.
Oof, yeah, that's a doozy of a question. I saw it in the chat and I was like, let me think about this while everyone's chatting. I mean, there is definitely, I think it just adds more data points to our understanding of life. We have four terrestrial planets in our solar system that we can really start to think about how life might arise, maybe add a couple moons in there. Maybe it's, 6 or 7 objects, but this adds, thousands more potentially. I will say I did a check today because this number changes every day. In terms of the number of potentially habitable exoplanets. And you can take that definition however you like. The number is roughly 50 to 60 planets out of those 4,000 plus planets that we found. And so that number slowly grows. But even among that subset of roughly 50 to 60 planets, they're primarily based off of the distance they are from their stars, the temperature that they might have. But those are very big constructs, and understanding the processes that might create a habitable planet is yet another problem in and of itself. So understanding, how weather might play a role, how the chemistry might evolve, not only in its current state, but over time. Another thing I'll add about M dwarfs is that their time on the main sequence of a star and the evolution of the star is much longer than our sun. And so how might life arise over that long time period? Is it going to sustain, or is it going to just be blown off, before we even have a chance to look at it? And so I think it's understanding the persistence of life, but understanding, how these different mechanisms that we understand fairly well in these solar system planets and applying it outward, but really just understanding the diversity of planets that probably exist out in the universe.
Yeah, I think one of my favorite things that we've learned just from looking for other planets is that the organization of our solar system is not the only way to do it. Until we had the detection of planets around other stars, and the first ones we detected were the weird ones, right? It's like if you think about when you go hiking, where your brain immediately wants to pick up like the really shiny white crystal of quartz in the middle of a lava field, right? Your brain looks for this crazy thing, and then eventually you learn to look for all the detail. But we found these giant planets very close to their star, which is not something that any of our models could explain. We had no idea. So it's completely shifted how we think the solar system's history, how our planet formed, all of that. It's just absolutely incredible. I wanted to make sure that we got to a couple of questions about life as we know it, but maybe distant, and is it talking? So for this, I'm gonna bring in my colleague Squeaks, and we'll go back to our background from Arecibo, and let's just talk for a moment about SETI. So the search for extraterrestrial intelligence and other things. People are familiar with SETI and that concept, and obviously there's a history of the Planetary Society with that concept, with contact, with this kind of stuff, the work of Jill Tarter. This is a really important concept, and it's really, really evolved with time. We think about it in this classic, okay, we used Arecibo to listen for a radio signal, but there's a lot more to it. And in fact, it actually is starting to touch on things we'll be able to detect that are much more systematic, perhaps, than the chance of detecting the signal that we imagine, in the era of Frank Drake. And so this concept of the search for extraterrestrial intelligence, or the way that we call it now, technosignatures, is a really important part of astrobiology. It's kind of a little bit of a separate activity. And one of the things that it's asking about that I think is really neat and critical is, does that ability to communicate actually evolve? That's actually an important question. We know on our planet, for instance, that dolphins have their own language, and it took us forever to actually detect it. We could hear the sounds, could hear it. And it was until we figured out information theory that we realized they could talk to each other, they had names for each other. This is an incredible discovery in our own concept of what life is like. And so I wanted to kind of spin away from just the traditional things that we think of with SETI and thinking about technosignatures. So a little bit back to Tiffany and atmospheric signatures. So there's a couple of papers now on what would the atmospheric signature of technology be, of the rise of a civilization. Some of the climate things that we've unfortunately done to our planet are actually detectable from space. And so the actual detection of another planet with elevated carbon dioxide might be actually this technosignature we're looking for, not exactly a radio blast from Stellar Radio 99.1. So Tiffany, could you talk about that a little bit?
I'm glad you brought it up. It's a really, I think it's a field that people have been thinking about for a while, but I agree, it's really blown up in the past, decade or so. I think with our understanding of how these technological pathways might be unique. One I can think of off the top of my head is related to what you just mentioned. If advanced civilization was using fossil fuels like we do on Earth, what amount of CO2 gas might be, what volumes of CO2 gas would indicate that, this is above what, an atmosphere and chemical equilibrium would produce on its own. And so that has to come from, factories or whatever. And so just thinking about, the advanced activities we have here on Earth, the advanced technologies, what are those signatures that might be imprinted on potentially habitable exoplanet and how we might be able to see that. It really, again, opens up a whole nother avenue of possibilities that even I can't think of.
Yeah, I thought it was a really great way of focusing on, we have these concepts of what we call like high risk, high reward. And often it's, if we're going to keep going back to this horrible cake analogy, it's ingredient focused, right? Okay, we found an egg, it's absolutely a cake, right? But this idea of system science that we all kind of mentioned when we talked about what astrobiology and life detection is as a field, it's important even for exoplanets, for this planet, for anything. Tiffany was just talking about atmospheric signatures. One of the things that might trick us actually is oxygen. So the classic thing that we talk about is that the Earth has more oxygen than it should, and it has it because of plants evolving the ability to create an atmosphere that we now use. And if you think about it now, actually, we know that oxygen is like horribly toxic. So if you just took it to a planet that didn't have it and dumped it on a living planet, everything would probably die, and eventually something might come up and be able to use it. But that's this whole connection between the history of our planet and what life did, the examples of that is a system science. It is putting all of those things together. And that's why it's hard to detect life, right? It's a very challenging question to answer. Betül, I wanted to pass this over to you a little bit because I wanted you to maybe just kind of share with people, how is it that you explore the molecular history? Because we're talking about technology for detection, what life was doing. How do you see the environment in the DNA of an organism that you're engineering in or working with in your lab? And I wondered if you might talk about the concepts and the tools that are used. The thing I think is really neat is the way that, like, we're all living in COVID-19 land, and we've been talking about labeling, right? The mRNA vaccines, we talk about CRISPR getting the Nobel Prize. These are actually technologies not so far and very related to what you're doing in your lab. Would you maybe speak about that as well?
Exactly. The mRNA vaccine is actually a great example. It is a result of our understanding of basic chemical architecture of life without altering it. So now imagine that if we actually figure out how this chemical architecture that acts like biology or life itself, if we figure out how that happens, imagine the possibilities that are waiting for us, aside from figuring out how life started, just because it is the biggest question, or maybe biased, but it's a pretty big question. So actually one thing that Tiffany mentioned was the sustainability of life. So that's where I will take it from. So we think that life is at least 3.8 billions of years old, right? So there's 3.8 billions of years of evolution on this planet. It is remarkable. And about 90% of everything that happened, at least at the genetic level, is wiped off. So we have about 10% data to work with to understand what happened in the past. All right. So first of all, we may have to accept that we may never truly understand what happened because we don't have a time machine. And I live with that. You know, it doesn't keep me up at night, but you know, I accepted that we may never truly know, and that's okay, as long as we understand the possibilities or come close to understanding it. So the way we work is that there are two phases of origin of life, or at least the beginnings. One is the tractable history of life. So how much can we use today's biology to track and bootstrap our way backwards in time to reach out to our last universal common ancestor, this hypothetical organism that we think all life originated from. We can, to a degree, track things. So we look at all life, you, me, a crocodile, a lizard, a snake, and then try to understand what we have in common, and then bootstrap our way back to the most commonality and subtract everything to understand what this early primitive organism might have. But then there's another side to this, because life started, our planet is 4.5 billions of years old. So that's a pretty fast time for life to flourish. Like we're talking 500-600 billions of years. So that's pretty fast. But that area is completely untractable. Okay, so whatever happened there actually created the chemical architecture of life that 3.8 billions of years later enables us to protect us from a pathogen, right? Because we figured out that's RNA chemistry, it's mod RNA, right? It's RNA-based vaccine, it's RNA world, really. We live in an RNA world today, if you think about it. And we cannot track that part of the history because it's completely untrackable. There's no record of it. This is, I think, why studying different planetary systems and moon systems in our solar system is important. And this is what I was trying to get at, the proto-Earth chemical conditions. So in a way, places like Mars or Europa give us that, you know, a picture of that untractable phase of Earth that enabled all life here. And I want to give another example from Star Trek. You may, if any of you are following that or fans of it, you may remember that there was an episode where you know, that we traveled back to a primitive planet, and we had to actually, we were seeing that how life may have started in this goo formation, and how Q was meddling with this goo, thinking that, oh, don't touch it. Well, you just ruined life, right? So we are talking about some sort of contingent system that, you know, we think that maybe those stars aligned, no pun intended, and maybe everything got, you know, together in such a way that life enabled, or did life originate on multiple times but did not sustain itself. So there are maybe a lot of contingency into this, and there may be a lot of differences into this. And our approach is two ways. We try to extract what we have today and travel backwards, but at the same time, try to create early chemical conditions. We do experiments, we do radiation experiments, we radiate mixtures of chemistries and with gamma rays, and try to understand, it's really, if you're thinking of Hulk right now, it is like that, but at the chemical level, and see how these radiation environments may impact early chemistries and enable structures that may look like metabolisms or networks that have some lifelike behavior.
It's pretty incredible. So we just have all these questions for other planets, but we're still figuring out our own. And so I really like it. I think the history of life on this planet is the history of ourselves, and is the history of our planet, our solar system, and the universe. So I just want to thank everybody. Thank our excellent panelists, Dr. Tiffany Kataria, Dr. Betül Kacar, and Dr. Heather Graham for joining us today. It's been an absolute pleasure. It's been an honor of a lifetime to be a member of the board of directors for the Planetary Society. And thank you, everyone, for joining PlanetFest 2021, and have a fantastic weekend.
Thank you.
Thanks to all of our speakers for sharing their expertise with us today. Please visit the exhibitor area to meet our PlanetFest supporters. Click on exhibitors in the far right menu to take a peek. Coming up is the next panel on doing science on Mars. Please click on the agenda to the far left and choose the session title to connect. Thank you.